Tēnā koutou. No ingarangi, no airangi o ka tupuna. Ko tami makaurau te whenua tupu. Ko riko na te kāinga. Kei te whānau wānanga o Waitaha taku mahi. He pukinga o te kuna nāhiri. Ko Sarah Wyse taku ingoa. Nō reira, tēnā koutou, tēnā tātou katoa. Kia ora. Oh, I should start my slides there. Kia ora. Thanks for the introduction, Tiffany. I'm a lecturer in the School of Forestry at the University of Canterbury, and I'm not a forester. I'd like to put that out there. I'm a plant ecologist, forest ecologist, and my interest is in our native forests and what we can do to best conserve them and get them back into our landscapes. And some of the research that I've been doing recently with some colleagues at Manaaki Whenua is just trying to get a handle on actually what do we know about seed banking of New Zealand species. Parts of the world where I worked at the Royal Botanic Gardens Kew initially, in this space, Europe's doing really well. They know a lot about their native species, about how to look after them, how to conserve them. New Zealand, we're still in our infancy in this space. Australia's doing better than us, unfortunately. So we're still learning. And so our bit of work here, which I'll be talking to you about today, was thinking about just trying to understand what we already know, where the gaps are, where we need to go into the future. And I should point out, this has just been accepted for publication, so hopefully it'll be out soon. Thank you. And I'll give it to Mel, give Mel a copy. She can send it around if anyone's particularly interested in reading the dry scientific paper. But I'll start by just explaining a bit about the seed banking process so we're all on the same page here. And we start by being out in the field, collecting seed and figuring out what species actually we've collected seed from. That seed comes back to the lab where it's checked. We look to make sure that the seeds are actually got embryos in them, they're filled, they're viable, they're not got little insects in there eating them out. We do some measurements on them. And then in the bottom left corner here, see my old colleague from Seed Bank, Sian, she's carrying a nice little bag of seeds into the drying room. And so the seeds are then dried, and this is at about 15% relative humidity. They're put in this room to dry out and lose their moisture content before going into the middle bottom there to the freezer at minus 20 degrees to be frozen. The final little picture there is indicating that those seeds are then monitored. So they come out, you check the viability, you see how they're going, and make sure that they're not losing viability through time. But, and here's the big but, not all seeds can cope with that process. They can't cope with being dried, and some of them can't cope with being frozen, which presents a bit of a problem if we're trying to store these seeds. So we can think of all plant seeds as being part of what we call the desiccation tolerance continuum. So on one end, we've got seeds which, like your typical crop species, your wheat, your barley, bean seeds and things, they'll really, really happily cope with drying out. And we call those orthodox seeds. This is a term that's come from early on when seed banking was developed for crop species, basically. So these are the easy ones. These are the ones they will be able to dry out, they'll be able to be frozen. So these are nice easy ones. At the other end of the spectrum, we've got a group that we call the recalcitrant species. They're the ones that are a problem and they're behaving a bit, causing some difficulties for us. These ones are desiccation sensitive. They do not like to dry out, and so they will die if we attempt to remove their moisture content. In the middle, so it used to be that we'd said, okay, there's these two categories of seeds. Life falls into two nice happy bins, but life often doesn't fall into two happy bins, and there's a mixture in the middle. And we call these the intermediate species, which if you treat them just right and you sort of coerce them a bit, you might be able to get them into a condition where they will be able to be stored in a seed bank. So on this diagram here, on the one end, we've got our examples from our crop species, our orthodox species. The other end's things like mangroves, cocoa plants, oak trees, and in the middle there's some coffee and some citrus, which, give them the right treatment, you might be able to store them. So our orthodox species, our desiccation tolerant ones that can dry out, we can dry them down to about 15% relative humidity. And as we dry them, we increase their lifespan. So when you go to the garden centre, you get some lettuce seeds, tomato seeds, they've been dried out in those little foil packets to help them survive longer on the shelf, so that you can then have them still viable to plant in your garden. But we can also then freeze those dried seeds, and freezing them also further increases their lifespan, so that you can hold them without trying to germinate them for a longer period of time, with them still being viable. Our recalcitrant species are a bit different. So they don't undergo what we call maturation drying. So your bean seeds, before they come off the plant, they've dried out a bit. They've gone dormant, and they'll be able to be shed and wait until the climate conditions are right, they'll germinate. The recalcitrant species, they don't do that. They fall off the plant, raring to go. I've got a picture for Kākano here. Raring to go, ready to germinate very quickly. So they're shed in a hydrated state. If you take that water out, you'll kill them. They're metabolically active. Their cells are doing their stuff. They're alive. They're raring to go. If you dry them out, they will die. So we can't store them in a conventional seed bank. We can't dry them down and freeze them. So we then did a bit of work thinking, okay, well, globally, how many plants are we talking here that fall into this recalcitrant category that are going to be a problem? We crunched some numbers, and we came up with a figure that we think about 8% of all the plants in the world, or the seed plants in the world, are likely to have these recalcitrant seeds. Most of those are woody plants, are trees and shrubs, and most of them are mature phase forest trees, so that the trees of mature old forest. They tend to have big seeds that are dispersed by birds or other animals. And they tend to, globally, there's a lot of these species in the wet tropics. So think the Amazon rainforest. The last line there, occur throughout the phylogeny. So that means that we've got species that are quite different from each other. We've got oak trees, we've got palm trees, we've got monkey puzzles and Norfolk pines, and big tall trees from the Amazon tropical rainforest like dipterocarps. But it's strong and conserved at low taxonomic levels, we say. So species that are closely related in the same genus are likely to be doing the same thing. So if you know what one of them does, another one's likely to be doing the same thing. We also know that the species in the world that are most likely to be difficult to store are those that are also most acutely threatened. And this is with habitat loss. This is those tropical rainforests where habitat loss is such a big concern at the moment. But you're here to hear about Aotearoa. So what about New Zealand? So this is what we were then trying to do with our bit of research. Is we went through the scientific literature, we went through various reports that have been published, trying to go through all our plants and find what we knew about each species. And the short answer to that question is we don't know a lot. So on this pie chart here, the blue area is our unknowns, and there's a lot that we do not know about our native plants. But there is quite a bit that we do know, nearly 30% we do know something about, so that's a good start. We've got to start somewhere. And about 21% of our native plants we should be able to store, of the ones that we know about, right? About 7.5% we know are going to be an issue. I said that globally, the recalcitrant species are more likely to be those that are most threatened. So we had a look we compared our native flora about the seed banking characteristics and looked at DOC's threatened species classifications. Do we need to be really worried here? Do we have a lot of really threatened species that we're not going to be able to store? And happily, we don't seem to have that sort of picture in New Zealand. We've got issues, we've got recalcitrant species that aren't threatened, we've got some that are threatened. There's no sort of strong suggestion that the recalcitrant ones are more threatened. So that's a good start. But what about some broader patterns? And similar to global patterns, our native species that are most likely to be a problem for storage in a seed bank tend to be trees. They tend to be bird dispersed and have large seeds. They also tend to be in warmer habitats. So our tall trees, big seeds, bird dispersed, particularly from further north, in those lovely rainforests and Watakere Ranges and Waipua Forest, that's where we're going to have some bigger issues. But we also know that there's a lot of small things we just don't know anything about. Interestingly, shrubs, we have a lot of shrub species, and nobody's been particularly interested in finding out anything about them, where I guess there's been more research interest, certainly by early foresters, in trying to figure out how to grow our trees. So a lot of work still to do. And we can try and make some predictions. So if we do a map of the country, we try and look at what species are in each region. We try and predict what the unknowns are going to do. We think that of our woody plants, 81% of them we should be able to store in a seed bank. 17% are going to be a problem. And that's actually quite a high number for a temperate region. So if we compare ourselves to Europe, to the UK, we actually have quite a few. We've got a lot of species that are related to tropical plants. And subtle differences, but if you're further north, if you're in Northland, if you're in Auckland, if you're in the Waikato, you're going to have more species that are going to be a bit of a problem than if you're down here where we are in Canterbury. So we can go back to our arrow, our little diagram, our desiccation tolerance continuum from the beginning, and plug in our native species that we're all interested in. If we look at the orthodox end, those that should be able to be stored in a seed bank, we've got quite a few species on there that we can be hopeful about. Our beach species, for example, tawai, tawhauroriki, tawhaurorui, they should be fine to be stored in a seed bank. Rewereo, kōwhai, mero matai, actually. Nico, despite the fact that plenty of palms around the world are recalcitrant, our Nico seems to be okay. Kaikawaka and presumably Kāwaka, we don't know about Kāwaka, but it's likely to do the same thing as its relative. Kaikawaka should be okay as well. Of the Myrtaceae, of our myrtle species, Putakau and Rata, Mānuka and Kānaka, those species with small wind-blown seeds, we should be able to be storing those okay too. On the other end of the spectrum, we've got a few of our other podocarp trees. We've got Kakatea tōtara. These are species that we know won't tolerate being dried out very well and therefore put into a seed bank. Tawa, and presumably its relative Taraire. Kāpuka, koikoi, pukatea. These are species that we need to be thinking hard about how we can actually conserve them outside of their natural habitats. In the middle, Kauri, Towai, Rimu, intermediate species that maybe if we do a bit of science, we can get things, might be able to figure out how to get them into a seed bank. But of course, Kauri has some wider issues around Kauri dieback and species associated with it, which we might need to be thinking about too. So I just spent a little slide on myrtle rust. I know it's a hot topic. People are quite interested in looking at our Myrtaceae species. And if we think about the family, Myrtaceae, globally, it represents about 10% of all the trees in the world, and about half of those Myrtaceae species are a problem. They are going to cause issues for seed storage because they are recalcitrant. It's too big for genera, Syzygium and Eugenia. And within New Zealand, it's those fleshy fruited species. So the ones, I said, Putakawa, Rata, Manuka and Kākano with the dry seeds should be okay. The fleshy fruits are going to pose a problem. And there's a lot of good work being done by other people in New Zealand, particularly Karen Vanderbilt from Wellington. She's been doing a lot of good work thinking about how do we, what ways can we conserve these species. Just to preempt some questions that you may have, if we've got a recalcitrant species, we can't put it in a normal seed bank, we can't dry it in a freezer, what can we do? Do we have any options? The answer is yes, with some reservations. There is cryopreservation is one option that people are using overseas. And so this is the preservation of seeds or embryos from the seed, or maybe some other tissues, in liquid nitrogen. But it's not a case of just getting the seed and throwing it in a vat of liquid nitrogen to snap freeze it. There's some really complicated techniques involved, which are very specific to each species. So it's quite time consuming and expensive to work out the methods for each species. And some people have been trying very hard to find these methods for swamp Māori, for Syzygium Māori, and that work's still ongoing. You then need to develop the techniques to restore the species, to grow those little seedlings up and restore them to their habitats afterwards. We also know that within cryopreservation, lifespans may still be low, but it could be an option for some of our critically threatened species if you have enough funding and research effort going into it. And as I said, there's some good people in New Zealand doing some of this work. So where do we go from here? We need to, as I sort of highlighted, there's a lot we don't know, so we need to be working to continue to build our knowledge, and we need to be working together to do this. Not all of our knowledge will be found within the scientific literature that my colleagues and I were reading through. There'll be mātauranga out there, there'll be a lot of knowledge held potentially within nurseries that people have developed protocols for species, so we need to work together to find these tools, build this knowledge, and make that knowledge accessible for everyone, so that we can all. I'd hope that maybe we could ultimately end with a nice database where you could look up your favourite species and find out what the seed storage situation is for it. We need to be working on those options for our recalcitrant species. And we also need to understand longevity of seeds in storage. So it's not just a case of it's an orthodox species, we can bang it in the seed bank, shut the door, and walk away. Seeds have different lifespans in storage. My colleagues at Royal Botanic Gardens Kew are trying to get their heads around why some species live longer than others in storage. And there's a lot of quite complicated data. Trying to find patterns is tricky, but we know that lifespans for different species will differ. So ongoing monitoring of those seeds we have in storage will be really important. I'd just like to finish by acknowledging some of the excellent people I've worked with on this research. Colleagues at Royal Botanic Gardens Kew, John Dickey and Aisha Farrouk, Peter Bellingham, Tom Etherington at Manaaki Whenua, Tom Carlin from Scion, and Marcus Rongawhitia Shadbolt from Te Tirama Whakamātaki and the University of Canterbury, who's doing, as he said, some fabulous work with our Coprosma species and with understanding how best practices for conservation. And final acknowledgement to our funders, the National Science Challenge, Naraka Taki. So thank you all. Kia ora.
Kia ora, thanks for that, Sarah. We've got a couple of minutes for questions, whānau, and so I'll read a comment which you may have a question on, a response to. We've got two, one comment, one question. So, in terms of the comment, you mentioned dry banking or freeze banking So it's knowing the environment and surrounding. As expressed, dark hold vessels are better than clear. Hine nui te pō versus papa tuanuku. These are what support and provide growth to these collected seeds. So it's making sure that those environments are reciprocated in the seed banking, climatised, dry or frozen storage, clear or dark holding vessels. Is that a reasonable summary? And do you have anything additional to add?
Well, I found Marcus's comment earlier about the different holding vessels to be really interesting. It's not something that the scientific literature has addressed yet, and that will be really interesting to understand further, so we can learn some mātauranga around that. So yeah, we need to more questions to answer.
Okay, kia ora. Try and reduce that 71%. A question here, do you know if anyone has actually tested how long Pahutakawa survives for using the orthodox methodology?
How long it does survive? I don't know how long it will survive in a seed bank. I'm pretty confident that it is held at the Millennium Seed Bank at the Robert Gardens Kew. How long it's been there, I'm not sure. But no, we haven't actively done any work looking at the time span in storage, and yeah, some work I'd love to do. Yeah.
Kia ora, thank you for that. One comment here that one of our whānau are targeting hino, kohe kohe, te reire, matai, kahikatea, and a few others for seed collection and propagation. Karawhiua koutou, karawhiua. Any other questions, whānau? Awesome, well. There's one in the Q&A. Hold up, we've got a bit of a lag between chat and the Q&A. So, to what extent does freezing increase the average storage time for bank seeds? If keeping the freezers running proves to be a significant ongoing cost for native seed banks, can this mahi be done without freezing? And it continues, sorry. Especially if we are regularly germinating our seeds to check for viability, or planting out for ecological restoration, marae planting, etc.
So the question was around how long, what's the benefit of freezing? Is that a
Sorry,
summary?
I'll back up a little bit. So to what extent does freezing increase the average storage time for bank seeds, and can this mahi be done without freezing them?
Yeah, I'll scooch back to my little cheat sheet. I had a note on that. So as a rule of thumb, if you're a 1% decrease in moisture content or a 5 degree decrease in temperature will double the lifespan. As a rule of thumb. So you will be able to extend the lifespan if you freeze it than if you don't. And for some species that will be more important than for others. But if you're regularly germinating those seeds, growing them up, and then taking new collections from those individuals when they've matured and put on seeds, then you can maintain that collection. And if you're regularly doing germinations, you'll be able to monitor how that viability is going, and if you're seeing a decrease. And we know that they'll truck along quite well without doing many changes, and then they'll suddenly start to go. So you should be able to see when viability is starting to drop off, and where you might need to start doing getting those seeds in the ground, and then getting seeds, propagating those individuals to get the next generation potentially.
Kia ora, thank you. Question from my nephew, actually, on behalf of Ngāti Koro ki Kaukura. Is there a way to store seedlings or slow down the growing process as a way of storing recalcitrant seeds?
Storing seedlings. Not that I'm fully aware of anyone doing that formally. I mean, a lot of our native forest trees do that naturally. That's what they do in the forest. They'll, like Kodi, for example, will germinate, and you get a little seedling bank that sort of sit there and wait for a light gap and where they'll take off. So yes, you could potentially have a go at holding some of them as seedlings to keep them a bit shaded, and see how that works. That could be quite interesting to understand.
Kia ora. Thanks for that, Sarah.